the opportunity to come here and speak to people. You're in a wonderful university and I really like it here. So thanks a lot for this opportunity, sir. And also I'd like to say, please keep this informal. I don't want to make this a formal lecture. I understand that you are all from different backgrounds. So feel free to stop me and ask me any question, any clarification, just now. So as I said, I did my PhD looking at adult genesis, and I did work on molecular mechanisms underlying the stem cell development. So I'll be telling you a bit about that. And the first question, what are stem cells? Most of you already know what stem cells are. In a nutshell, stem cell is a cell that can turn into any other cell type. That is a stem cell. And what about these stem cells? Why is there so much hype about these cells? The hype is because they may represent the next breakthrough in therapeutics. Drugs have been one era of therapeutics, stem cells and cell-based therapies making the next breakthrough. And in fact, recently, you may already know, Shinya Yamanaka was awarded the Nobel Prize for induced pluripotent stem cells. Stem cells are not they're not an elusive topic. They are present, in fact, in high amounts in the early embryo. So, stem cells early on during development are there and they are totipotent, meaning they can turn into any and every cell type. Naturally, that's what the stem cell means. But as the animal progresses and gets mature, this pool of stem cells reduces because there is organ formation. Obviously, you don't want your skin to turn into your muscle and your muscle to turn into your skin. So there is cell-fate specification and you want segregation so that there is organ development. But in that process, you also lose the number of stem cells, which is alright under normal physiology. But when you need it for repair or for regeneration, not all organs have the same regenerative capacity. Your skin, when it is hurt, there is wound healing. Skin cells repair themselves very fast. Same for liver. But that is not the case in all organs like the heart muscle or the brain. And within the adult, there are very limited niches in which stem cells exist. For example, they are there in the blood system, in hematopoietic stem cells. They are also there in the brain, and I'm interested in this pool of stem cells. So, like I said, these are not totipotent. That's very early on. Later on, when the animal progresses and gets mature, you have pluripotent cells, meaning they can turn into multiple cell types, but not all. So, in the brain, people have isolated, and I'll get into that in a bit. They've isolated stem cells which can turn into the 3 neuronal lineages, meaning 3 different cell types existing in the brain. The neuron, the astrocyte, and the oligodendrocyte. And each have their own function, but there is particular emphasis on new neuron production because these are the fundamental signaling units of this organ, the neurons. And as you know, with brain damage, there are many irreparable brain injuries where people cannot recover because there is hardly any repair in this established circuit. So, I've already sort of said this why question about brain repair. So once you have damage, can these stem cells be recruited? Can we induce stem cells to try and repair the damage that may occur in a stroke or in an accident or even in Alzheimer's and genetic diseases? Another interesting thing about new neuron production in the adult nervous system is that there was a dogma in neurosciences for a very long time. No new neurons. Neurons cannot be produced after birth. And that makes instinctive sense. You have a very complex circuitry and it cannot keep incorporating and investing in new neuron production. How will these new neurons get integrated? So this was thought to be the case for a very long time. And only in the last 30 years has this dogma been overturned and people have found stem cells to continuously make new neurons in the brain. So here is the cartoon of an adult brain of a mammal, and this is a sagittal view, meaning it's cut this way and you're looking at it from the side. So this is the nose, the nasal region, and this is the back. Stem cells have been isolated in vitro, meaning adult tissue from mice and rats and other mammal model systems has been taken, dispersed, and cultured in the dish. And you can see stem cells. You can see that there are cells which can turn into other cell types, which can make new neurons, make new oligodendrocytes. So the adult brain harbors stem cells in many regions, like the olfactory bulb, which is used for smelling, in the lateral ventricle, cortex, which is thought to be a very, very human and very higher mammal area. Also in this spinal cord, which is very important in repair for injuries and related to accident. So all these places have stem cells, but they can only be found when you isolate them in the dish. They are not continuously making cells in vivo, right? So now there is the big gap, not only between why do we see them when you take them out of the natural environment. That is one gap, and the second is there's a lot of technology. People have made layers of heart muscle. People have made even neurons in the dish. But there's a big gap to bridge when you have to put them back into the brain, back into the spinal cord, back into the heart in the live animal, human beings. Why doesn't it work? Where is the problem? So in this case, what we wanted to do was to look at endogenous stem cells. So in these two colors, so here in blue and here in green, are two regions where there is endogenous production of new neurons. The rest of these areas, only when you take them out into the dish. And in these two areas, you have continuous production of new neurons throughout life in mammalian systems, including the human. And this has been found in humans in postmortem studies. So these areas have new neurons being produced continuously till death. This keeps reducing over time, but it still occurs. And I'm particularly interested in this area. And understanding these stem cells, how they integrate, what are the molecular mechanisms, will allow us to also ask, can we use the same molecules and apply them to other stem cells in the dish and use them to manipulate them, to put them back, get them to integrate into the brain. That's one. And second, also, can these guys be recruited for repair? Can these stem cells be transferred to areas of brain damage? Can we manipulate them with techniques and tools like viruses or drugs, injections, to see if they can be of use in the same situation? So here's again another schematic. And when you look at a coronal section, you look at it this way, you look at it from the front, it looks something like this. This is the structure of the hippocampus. I'm not going to go into an anatomy lesson because that is not relevant and you don't need to know this. But what would be useful for you to just sort of keep in mind is that in black here is a set of packed cells. That's called the granule cell layer in the hippocampus. And this is all made already during development, right? And what is left, these stem cells reside here at the border, in this red region, called the subgranular zone. So the stem cells reside here. They go on to divide, mature, integrate, and always in something of a similar fashion. So I'm not sure if you know the basic morphology of a neuron, but basically a neuron has dendrites, which are something like antennas, where they receive input, and then it sends an axon out to its target cells, many cells. And during development, there is a whole set of transcription pathways. There are morphogens, there are growth factors, which allow cells to grow and form and connect in a certain way. That's how everybody's brain is basically the same. How do you always achieve the same connectivity? More or less. Is because there are entire programs that allow the brain to form in a certain way. And these guys are now born in a pre-established circuit. There is already a brain, there are no more gradients. So it becomes really interesting to ask, how do they know to always send their dendrites in a certain place? How do they know to send their axons and connect to a certain place? So here we have looked at endogenous pathways and molecular mechanisms that may allow these cells to grow in a certain way and what is important for their integration into a pre-existing circuit, which is something you need even for a cell that needs to be transplanted. How will it integrate? So again, if you zoom in into this region here, you will see something like this. So the black area that I told you was this packed cells or something like that. And here is a timeline of how these cells grow. So this is the prototypical neural stem cell in the hippocampus. It looks something like this. It hardly divides. It self-renews, meaning it symmetrically divides, gives another cell which will then go on to become terminally a neuron. So that it maintains its own pool. And these cells, once they divide, between 3 to 10 days, they change their morphology completely. They become small, they start dividing, and then the ones that survive, meaning not all of them survive, most of them are eliminated, only less than 5-10% make it to a mature neuron, go on to arborize, they send the dendrites, they send their axons out, and finally they look something like this. And I'll use the word dendrite and dendrite arborization. Arbor because it's like a tree. Arbor means tree, and it branches like a tree. And these are all interesting and important parts of connectivity, how these cells get into the circuit. So, apart from what I mentioned to you already about stem cells, of how looking at pathways is important, this process alone, hippocampal neurogenesis, is not just a static process. I told you the dogma got overturned, neurogenesis continues throughout life, but it's not static. It's highly responsive and regulated. It's amazing in terms of the number of papers over the last 10 years that have shown what all regulates neurogenesis. Enriched environment, running, activity, stimulation by a special environment, all of them increase neurogenesis. And these are studies mostly using mice and rat models. So they increase the total number of neurons. Learning increases it. If you put a mouse in a maze, force it to learn, that increases neurogenesis. So that's good news for everybody here. We're all having more neurons. Antidepressant drugs, they increase new neuron production. So clearly this is not just a static process. It's highly responsive and probably takes part in adaptation, plasticity, and responding to the environment. It also has negative regulators, trauma, social stress, physical stress, models of depression, all dramatically reduce neurogenesis. And it's still not clear whether this is an adaptive function or a causative relation. Does a loss in neurons promote a depressive phenotype? So that is still being explored. But people know that these neurons play a direct role in learning and also in emotional responses. So clearly this is also a very, very dynamic process at the interface. And the question remains open. What happens on the outside of the organism? How does that get translated to an effect on single cells? That's a very interesting question. How does stress on the outside or running or learning, something so abstract, influence single cells and entire cellular processes? And here it becomes important to ask, what are the molecular mechanisms? Because eventually the cell experiences the world through its receptors, through the input it gets, and finally molecular pathways which influence its behavior. And there are many molecular pathways out there which one can explore and people are exploring. And I chose to focus on the low GPases. I don't know, have you heard of low GPases? So they are usually, people don't think about it as specific to cells or to neurons. Throughout the body they are present and they have a role to play. They are extremely important molecules. They basically act like binary switches. They are GTPases, so they are either bound to GTP and they are in the so-called on state. When they are bound to GTP, they bind to downstream proteins, which then bind further downstream proteins and finally affect several processes in the cell related to cytoskeleton, microtubule dynamics, cell movement, and others also. And once they hydrolyze the GTP, they go back into the off state by being bound to GDP. And these are controlled by these proteins whose name you do not need to know. But essentially you need to know that they integrate upstream signal and cause downstream effects. This is a very simplified version. There's an even more simple version. So here is something of reality, but also it's much more complex than this. You have upstream signals like growth factors. I don't know if you've heard of nerve growth factor, brain-derived neurotrophic factors. These are brain-specific factors, but they are equivalent to any growth factor also that you would have in any other system, growing plants. So these affect molecules that turn these proteins on and off, and these are three major members of the ROGPase family. They also interact with integrins, which interact with the extracellular matrix, meaning directly with the environment. If a cell or a neuron has to grow, you know the brain is a very dense organ, right? Most of us haven't touched the brain, but it is a very dense organ. And for something to grow, it has to start digesting its surrounding, it has to push and pull its way through. So the extracellular matrix has to be dealt with when there is growth and movement of the cell. There is also G protein coupled receptors which also affect these proteins. And as you can see, downstream is also rather complex. And there's a lot of overlap. And finally, what is the take home is that these molecules are important for many things, but especially the cytoskeleton. And I focused on these two guys, RAP1 and CDC42. RAP also has many 2 and 3 and other sub-members of the family, but RAP1 is what I was interested in. And as you can see, they have a lot of overlapping functions. And that is an interesting point to remember because what I'll show you is that when you look at it and its function in vivo, you'll see subtle differences. So all molecular pathways have subtle differences in how they affect the final behavior of the cell. So exactly what we asked was, what are the roles that these GPCRs play in adult organogenesis? And my experimental strategy to approach this question was first deciding on a model system. So we have in vitro model system as well as in vivo. But because the main thrust of my work was in vivo, which is where the real questions lie, I'm going to focus only on this for the purposes of this talk. But we also have adult hippocampal NSCs isolated from adult mice. So you take and sub-dissect the hippocampus of an adult mouse, and then you mash it up, and then you can isolate cells and grow them. And they really grow for multiple passages. That's what makes them stem cells. And then you can force them to differentiate into different cell types. So we did a lot of work with this, but I'll mainly focus on my work with transgenic and wild-type mice, looking at the in vivo roles of these proteins. And how did we ask how CDC42 and RAP1 plays a role? There are different ways. One is to knock something down. So you lose the protein and then you ask what is happening to the cell. And one way is to increase its function. So here I knocked its function down. And I'm mainly also again focus on these two for the purposes of this talk. But there's pharmacological inhibition of these. You can also knock it down. You can overexpress a dominant negative form of this protein, which is permanently bound to GDP, meaning it's in the off state. And also you can use transgenic mutants where you can just delete the entire gene from the DNA. And then you're looking at deletion of these genes in a live animal. And our readouts, if you remember from that scheme, the stem cell goes on to divide, it survives, it matures, becomes a neuron. I sort of just asked those questions one by one. What happens to proliferation? Does the cell divide more, divide less after loss of this protein? What happens to the neural cell fate? Is the total number of neurons less when you knock this protein down? What is the structure of the cell? And how do these neurons now integrate without these proteins? So like I said, I'll be focusing on these aspects. So coming right to the first question, is proliferation altered? Now I started by knocking this protein down using a transgenic animal. I'm guessing most of you are familiar with how transgenic animals work. So in vivo, what you have to do is start out with parent mice, like you have to do also with plant, breed and cross. And you might have already heard there are a lot of whole knockouts. About 10-15 years ago, people made entire knockouts, knockout of RAP1, which died already embryonically. So you could never take that animal to adulthood to ask, what does RAP1 do in adulthood? Because there were lethalities associated with the loss of this gene early on. These are called whole knockouts. What we now adopted was conditional knockout, meaning you can choose to knock out this protein where you want it and when you want it, using very smart promoters. So what we have here is a 3 recombinase, so the 3 recombinase excises out certain DNA sequences. So 3 cuts at LOXP. LOXP is the name of the site where 3 recombinase cuts. So what you would want is in one parent mouse, your gene is flanked by LOXP sites. And if this were to see a 3 recombinase, it will cut it out. So then you have deletion of this gene from the DNA permanently of that cell. Now you don't want the whole organism to see the 3, so you have the 3 recombinase expressed only under the promoter, which is active in your set of cells. So I'll go to my scheme, something like this, where nestin is a gene which is active in stem cells of the whole body. So now if I were to use nestin and cross it with this mouse, only in all the stem cells will the Cree go and cut off my gene, CDC42 or ACT1. But the problem with that is that, so here you get spatial control. It is only restricted to stem cells, but I don't get time control. I don't get to say when. It's already embryonic development in the brain, nestin is active. So people came up with even smarter tools. They fused Cree with the estrogen receptor. And the estrogen receptor, as you may know, is actually cytosolic. And for the Cree recombinase to work, remember that? Yeah, okay. Cree recombinase, Cree recombinase cuts at certain LOXP sites. So it cuts a certain sequence of DNA. Now if you take your gene of interest and put both of those LOXP sites, the Cree recombinase will come and cut them. If it's there in the nucleus. If it's there in the nucleus, it will see them and cut them, and then you have your gene gone. But if you have the Cree everywhere, all over the body, so all over the organism, there will be deletion of in the skin, in the eyes, in the heart, and the animal dies in our case. Because these are very important genes earlier. So you don't want this to be everywhere. So what you do is, you say, okay, I want Cree expressed only at in certain cells. For example, there is a neuron-specific promoter, a neuron-specific enolase, which is expressed only in all neurons. Not in the skin, not in the eye, not in the heart, no, only in all neurons. If I were to put Cree under that promoter, so only when the transcription factors that drive neuron-specific enolase will turn this on, meaning you'll have expression of this only in the neuron. So you will cut out your gene only in the brain. So what we did was even went one step further. We went to the stem cell. People know from their work which promoters are active in stem cells. So people know that certain things are active in stem cells, certain things come up and go down. So we know that nestin is a gene that is expressed in stem cells. But the problem is that it's expressed also during development. So brain has a lot of stem cells when the animal is being born. So how do you control for time-wise control? How do you say in the brain and only in adulthood? That's the next level where people came up with very smart tools. They fuse this Cree. So DNA excision requires its presence in the nucleus, is that correct? People, they made another fusion protein. So they modified it, fused this with the estrogen receptor. Estrogen receptor is present in the cytosol, not in the nucleus. So that caused rerouting of this Cree recombinase only in the cytosol under default conditions. So then you have a stem cell which has Cree only in the cytosol. So your gene is safe, it's not being deleted. It gets deleted only when the Cree recombinase goes to the nucleus. When does estrogen receptor go to the nucleus? When it binds to estrogen. So what we do is take an agonist. So you take a drug which mimics estrogen's properties in binding to this receptor and you inject the animal when you want it. You can choose to inject it in development, you can choose to inject it in adulthood, you can choose to inject it when the animal is 2 years old, which is very old for a mouse. So this gave us temporal control also. So here, now I go to this key. It's not more complicated, it's the same. Nestin expresses my Cree recombinase, which is cytoplasmic. And here are the exons which I would like to give you. So you have two parent transgenic mice, you cross them, you get them, and then once you have those progeny, you inject them tamoxifen, which is an estrogen receptor agonist. And then you cause deletion of these genes, only in adulthood, only in stem cells. Third question, how do you see those cells? Stem cells are very few in the adult brain. How you see them is by another gene which is already there in this animal. It's called a reporter. So what you have is a fluorescent protein which is preceded by a ubiquitously active promoter called the ROSA promoter. But the thing with the ROSA promoter is that there is a stop codon, meaning under normal circumstances there is no fluorescence. Only when the Cree is active and the stop codon is gone, you will have permanent driving of the fluorescent protein. So then you have permanently tagged that cell which has seen the Cree recombinase. So using these kind of genetics, you can achieve very good in vivo deletion of the proteins and of the genes that you want, and look at very specific readouts in the cell. So this is what we have. And then, going back to the original question, is proliferation altered? Do the cells divide less? How do you assess division in vivo? One way is to give it a thymidine analog. So what I did was I took my transgenic mice, which have all these genes, injected them tamoxifen, waited 10 days, waited 30 days, and waited 90 days to sacrifice them. To see at what time point will I see an effect. Is there a delayed effect? Is there an early effect? And 24 hours before sacrifice, I injected them with EDU, which is a thymidine analog. Are you guys aware of what the thymidine analog does? Somewhat. Okay, so basically when a cell is dividing, instead of the T that pairs with the A, you have this EDU that goes in there. So you have labeled, and you inject the animal systemically. And then you have BRDU EDU being incorporated everywhere, not just in the brain. But that is not my concern. And then you can label these cells by doing immunohistochemistry. So you can put an antibody against EDU and then put a fluorophore, so then you know that cell was dividing 24 hours before sacrifice. And then you want to ask at different time points, is this process altered? So my conclusion is already here, so you already know the thrust of the result. And here what we have are confocal images. So with confocal microscopy and immunohistochemistry, you can put two channels and go to a single cell level and ask, this green cell, which has yellow fluorescent protein, is the one which has the deleted gene. Was that cell proliferating? Yes or no? You can ask that by looking at the red channel, which is not very clear here, unfortunately. But you can see it here in the red perhaps. You can zoom in and do this single cell analysis with the help of confocal microscopy because it allows elimination of out of focus line. And we found that deleting CDC42, so in these CDC42 conditional knockouts, there was a reduction in proliferation at both the 10 and the 30-day time point. But the total number of cells were only marginally reduced, not significant. In contrast, RAC1, which has similar functions and thought to have overlapping pathways, has no such effect. And this is the same B structure that I showed you in the cartoon earlier. It's like this, and the rest of the thing comes like that. So with the loss of RAC1, there is no influence on proliferation. We next went on to also ask what happens to neuron production. Is there an effect because of proliferation also on neurogenesis? For that, you have to do immunohistochemistry and stain for a neuron. You have to look at the knockout cell and ask, is it a stem cell? Is it another kind of cell? Is it a neuron? And again, with that kind of single cell analysis, here I stain for SOX2 and GATP, which are stem cell markers, like nested, and ask, okay, is it positive for these two? Is the stem cell population changed? And we found that the stem cell population doesn't really change, but the percentage of neurons goes down marginally. Significantly, but it goes down significantly. So it goes down corresponding to how much there was a reduction in proliferation. So proliferation was less. It went from 6% to 2%. Similarly, neurons go down. And this is specific only to CDC42. So that tells us CDC42 is important for proliferation as well as production of new neurons. So we've already covered these two readouts and we know what gene is important for what process. The next question, the structure of the cell. Is the morphology changed? Is the arborization changed? And for this, I want you to take a look at these. It's very difficult to follow the whole cell. I made a cartoon of the cell where you saw these arbors and all these spines, but you cannot see that with this tool. So what we needed was a stronger driver for fluorescent protein. And what we used for that is a retrovirus. So this is another great set of tools that exist already outside. And we didn't have to make these ourselves, make the gene, but we had to make the virus. So a retrovirus, as you know, affects only dividing cells. It can only get into the nucleus once the nuclear envelope has broken down. And you know what a virus does, right? It infects a cell, takes its DNA, integrates into the host genome, or RNA, and then converts it into DNA, and then causes reverse transcription of its own set of genes, and then causes budding. That's how an infection with the virus happens. People have utilized this and made these viruses transcription incooperative, which means it can come into the cell, it can integrate into the DNA, but it cannot cause transcription and release of of further viral particles. So no tertiary transcription. Only whatever virus you put in, you will infect the cell. And that is a great way to put your DNA of interest into that cell. So what I did was, I used this viral strategy and combined it with transgenesis. So we have those flocks of mice, if you remember, with the lost P sequences. And instead of taking them and crossing them, which we already did, I put a virus expressing CRE. And so this CRE recombinase, which is virally transfected, will go and cut out the lost P sites and cause gene deletion in single cells. The advantage of this, I'll show you in a bit. I'll show you right now already. Is that you get cells which look relatively better. And a quick recap of overview of what it is that I did. So we injected the virus and we waited at 2 different time points where the cells are more mature and you can quantify their morphology. So again, red cells. So here you have an RFP expressing cell, that's a control cell. And you have a CRE expressing cell, which will cut out your gene of interest, and that's a mutant cell. So plain red cells are control cells and double labeled cells, green and red, are your mutant cells. So you can control for injection, you can control for animal variation by looking at 2 cells beside each other. One is a knockout and one is a control. And at the 15-day time point, what we see is again that CDC42, loss of CDC42 causes a dramatic reduction in the arbalization and the branching of these neurons. But loss of RAC1 does not cause such an effect. So again, it appears that CDC42 is important for not only proliferation, but also the morphology of the cells. RAC1 seems to be dispensable. Look at the 30-day time point, and there's something interesting that happens. Both of them have an effect. And here you can see much better. You can see here that a red fluorescent cell is very nicely organized. In contrast with here is a mutant cell which has the CRE and is labeled RFP, is rather not so nicely organized. And this is a quantification of total dendritic length. So using image analysis software, you can label all the neurites of each and every cell and look at the total neurite length. So that is the dendritic length. And you can express it as a cumulative percentage to see if there are a shift. So here I saw that there are far more shorter dendrite neurons in my knockout condition. And another readout of the growth and maturity of a neuron is how much it branches. And that can also be easily done using a software where you sort of put an imaginary dot here, and then there are concentric circles. And you analyze at what length are there crossings in the circle. So if there is a lot of branching, you have more and more crossings in the outer circles. And the graph looks something like this. So this is number of crossings and at what distance. So in both parameters, we find that loss of RAC1 and CDC42 by the 30-day time point influences the morphology of the cells. And this is different from the 15-day time point, where only CDC42 but not RAC1 influences. So now I'll come to the last point and then quickly do a recap, because I understand this is sort of precise conclusions that one can draw from this. We wanted to look at the integration of these cells. How do these cells connect with other cells? Do they receive input the same way? Do they also put their output the same way? And one way to look at that is to look at dendritic spines. Again, this is a blow-up of that cartoon that we saw previously. At the 30-day time point, cells are not smooth. They are like this, somewhat of like cacti. They have these dendritic spines growing up. And these spines are very important for receiving excitatory input. So here you can see a spine is the part of a dendrite which specializes to be a post-synapse. It has a high density of receptors, of post-synaptic scaffolding proteins, which allow signals to come through very fast and get translocated into the dendrite and then the neuron cell body. If it's all diluted in the dendrite, it seems that that's not a good idea. And here is the presynapse that would connect with postsynapse. Now, what I want you to remember here is that there are different types of spines. There is a thin spine, which is like a philopodia, which fishes around for input. There is a mushroom spine, which looks like a mushroom. And why is this mushroom spine interesting? Is that you can see it has a very large head and a small neck. And this seems to be related to the maturity of the spine. So So the more mature the spine is, the more input it gets, the larger area its head develops. So what we did was we first looked at using semi-automated analysis in 3 dimensions. We looked at the spines of virally labeled cells, where I've used dominant negative approach. And here we look at the spine density, so the number of spines per length of dendrite is not ordered, which is a good sign to some extent because these proteins are so important for everything that one would be afraid that you lose all input altogether, but that is not the case. What you do lose is specific input and maturation of spines. So there is a reduction in the percentage and the number of mushroom spines only with both loss of CDC42 and RAF42. So we also found that these proteins are important for the integration of these neurons. So here's a set of conclusions that we can draw from this. And this is not just based on what I've shown you. We also have some other data in vitro that very much complements what we found in vivo. So CDC42 seems to be important at all the different stages. It's important for proliferation of cells, it's important for early maturation and also the late maturation and integration. In contrast, RAC1 seems to have a rather late role to play. So 2 members so similar, the same family of GTPases, have distinct and stage-specific roles to play. And that is what we essentially found. And these are novel regulators of gluconeogenesis. And it is possible that if one wants to now take this, use viral technology with exogenous or endogenous stem cells, progress can be made on that front. So with that, I'm already going to conclude and thank the people that we had. I did my work in Sebastian Escobar's lab in Zurich, and also our collaborators. And we published this work in Journal of Neuroscience recently. So that was good. And I actually have a lot of other slides in backup, provided you guys give interesting questions. So I've kept them in the back in case you want more explanations. There's a lot more data. But I thought I could keep this more for a general question and answer session about neuroscience or neurogenesis in general, not necessarily about